Dr. Asta Arendt-Tranholm is our guest today. She completed her PhD at the University of Nottingham in England and then came across to the United States to begin work with Dr. Ted Price at the University of Texas at Dallas. She is the first, but not the last speaker. As you can see, we've got a whole lineup ready for us to take advantage of. If you have questions, and I hope you do, please submit them in the question window. At the end of Asta's seminar, we'll have time remaining for questions, and we'll address them all at once at that time. Okay, so Asta is part of the Precision Human Pain Network. This is a HEAL initiative that probes the genetic and cellular basis of pain. And the entire consortia of data related to the Precision Human Pain Network will be available on Sparc. So this is a growing data set. And with that, I'll hand it over to you, Asta.
Thank you so much. It's such a pleasure to be here. Let me see if I can share my slides and we can get started. That should hopefully make my slides visible to you all. Thank you for the really kind introduction, Sue. As she said, my name is Asta. I am a postdoctoral research associate at the University of Texas at Dallas in Ted Price's group on pain neurobiology. And I'm going to be talking a little bit about my work today on characterizing novel RNA isoform expression patterns in the human dorsal root ganglia and how we collected all that data and carried out that work using long-read sequencing. I've been a postdoc in Ted Price's group for going on 3 years now, and this has been ongoing work, but also work that we published last year that I'm really excited to be talking about and sharing with you today. So, given that we are a bit of a diverse group, this is the overview of my seminar. I'm going to start with a general overview, some background, and an introduction to my talk. And then I am going to be going into the data procurement from long-read sequencing specifically. I'm going to be going over the analysis that we did and the validation of short-read sequencing. And then I'm going to be talking about the interactive platform that we generated and how we made our data publicly available. And then I'm going to have a brief summary, talk a bit about the future of this work. And then at the very end, there's going to be a little teaser for a different data set that we're also working on. So I'm excited to talk about all of it with you today. So to begin with, what is splicing? Splicing is a post-transcriptional mechanism of the pre-mRNA that is carried out by the spliceosome within the nucleus. So in this story of flow of information from DNA to RNA to protein, we are in here between the DNA and RNA. A pre-mRNA has been produced through transcription. And while it's in the nucleus, it is spliced to basically remove these intronic non-coding regions to produce a mature mRNA, which can then be transported outside of the nucleus to the rest of the cell for translation to produce a protein. Basically, splicing, or the process of alternating to splicing, is the removal of the intronic regions to just leave the exons, which are the coding regions of the RNA. And it's been found, this was a really nice review, that this process can be done in a few different ways in eukaryotes. The most common that we see is this cassette exon. So basically, an exon, a coding region, can be included or excluded to produce two different transcripts that, when translated, will produce two different proteins. And to just go back to this previous figure, which I just think is really nice, it's been found that this process can actually occur in a few different ways to create a multitude of different isoforms from the same gene. So we don't just have the cassette exon where the mature mRNA is either including or excluding an exon. It can be uses of alternative splice sites at the 5' or 3' region of an exon to include different parts of an exon in the mature mRNA. You can also have an exon spliced out entirely, of course, or you can actually have intronic regions be included as well. So regions that were otherwise considered non-coding be included in this final mature mRNA. And finally, there's this example of mutually exclusive exons. So exons that are present in the pre-mRNA, but are never observed in the same final mature mRNA. They are only included one or another in the final mature mRNA. The functional implication of splicing is actually immense. From the 1990s, splicing has been characterized to have an immense functional effect. This paper specifically characterized the splicing of the calc-A gene into either calcitonin, which was expressed in thyroid cells, or a calcitonin gene-related peptide, which is expressed in the nervous system, so CGRP. They're both peptides, but they have entirely different functional roles. So not just expressed in different cells and different tissues, but have entirely different functional roles in those tissues. In the thyroid cells, this peptide plays a role in calcium concentration within cells, whereas it's a neuropeptide in the nervous system, which we now know to play a pretty important role in the pain processing pathway, and specifically in migraine. Since then, splicing has been shown to play a number of different roles. This paper from Christine Holt's group was really interesting in showing not just that there can be different transcriptomes in different parts of cells, but what they actually showed in this paper, or in this project, was that there was much more transcriptome or isoform diversity in one part of the cell than there was in another, which can, of course, have an implication for the final proteins that are expressed in different parts of the cell that play different roles in different parts of cells. There's also been quite a few different stories that have come out now from different groups showing how individual genes can be transcribed into or can be spliced into different isoforms that have these functional and sometimes opposing roles. So the Robo3 gene is a really interesting story here around two different isoforms having functionally opposing effects. So Robo3 plays a role in axon guidance. And there are two different isoforms of this transcript that can either guide attraction or repulsion of axon guidance. In addition to this, this paper from Dan Lipscomb's group showed how different isoforms expressed in specific cells, specifically in neurons, had a different functional implication for the calcium currents that drove pain sensitization, also in the nervous system as well, of course. And finally, this is a story that we're going to touch upon a few times in this presentation, but the WNK1 gene, the Wink gene, Wink1, is a gene where there has been a unique isoform that's been characterized as expressed within the peripheral nervous system. And we'll come back to the role that that specific isoform plays. So a multitude of functional implications of splicing have been characterized, but in order to understand how these isoforms might have a functional role or how their impact may change, we need to know what isoforms are expressed basically at baseline or in a different, in a given tissue at a given time. Once we know what those isoforms are, we can then start to characterize how that transcriptome or the spliceosome may change in a given phenotypic condition. What's worth noting is all of the research, those three instances that I just spoke about, they were characterized in the rodents transcriptome. And what we're really interested in Price's group is working with human tissues. So I want to talk a little bit about the human tissue procurement. First, the tissue that I'm particularly interested in and that a lot of us work on in Price's group is the dorsal root ganglion, or the DRG. It is part of the peripheral nervous system. These ganglia sit outside or just outside the spinal cord. They're basically involved in transmitting sensory information from the periphery all the way through to the spinal cord and up to the brain for us to experience sensations, including pain. These DRGs sit at each level of the spinal cord, and all the cell bodies are within that ganglia itself. I just want to touch upon a really important collaboration for our work, which is the work with the Southwest Transplant Alliance. So the Southwest Transplant Alliance is how we get access to the tissues that we work with, which is tissue from human organ donors. Most of us in Ted Price's group are actually on donor recovery teams. So we go out to donors sometimes 2 to 3 times a week to collect tissue. And as you can see, our postmortem interval is 2 to 4 hours. This basically means is that we come out just after all of the organs have been recovered for transplant. We collect the tissue that we recover and take back to the lab. So it's a really short postmortem interval, which means that the tissue is in really quite good condition. There's a number of different tissues that we collect now. This has changed a bit over time. So we're not just collecting the spinal cord and the DRGs, we're collecting a few different ganglia, as well as some peripheral nerves. This tissue is either carried back to the lab on wet ice, so it's kept alive in cerebrospinal fluid, and we can culture these tissues or cells from these tissues, or it is snap frozen in dry ice. This is basically done to preserve or to stabilize the RNA, or for us to section the tissue, for example, when we get back to the lab to retain and explore the morphology as well as the transcriptome. In this case, for this study, what we did was extract really high quality RNA from these frozen DRG tissue from a few different human organ donors. The goal being, or the aim of this project being, to characterize the isoform expression patterns in the human DRG. To do that, we needed to obtain full-length transcript recovery, which is what facilitates this isoform differentiation I was talking about previously. Specifically how we can parse out the specific patterns of how exons appear to form unique isoforms in the human DRG. To do that, we used PacBio iso-seq long-read sequencing. And I just want to briefly touch upon the differences between long-read sequencing, which I've now mentioned a couple of times, and then short-read sequencing. Short-read sequencing is probably the more common type of sequencing. So when people talk about RNA sequencing, this is usually what they refer to. The difference being the way that the sequencing is done is that you collect snippets of a transcript. Usually it's 75 to 200 base pairs in length. And then throughout your analysis, what you're doing is mapping these small snippets of sequencing to a reference genome or reference transcriptome to identify which gene you've identified this snippet of transcript from. The missing information here is basically that you're not able to tell which exact order these exons are appearing in or whether they're appearing together. So you won't necessarily know if exon 2 and exon 7 are coming from the same isoform. You'll just know that the gene with this isoform has been identified. Long-read sequencing, the unique part is that you can obtain these, as the name suggests, long reads, sometimes thousands of base pairs in length, which is in fact what the transcripts are in the human transcriptome. There are many thousand base pairs, so can be up to many thousand base pairs in length. And this will allow you to retain this information regarding the exact pattern in which the exons appear, and as such, identify specific isoforms. The way that PacBio long-read sequencing works is that it is a circular consensus sequencing approach. Just super briefly, I am not an expert on this specifically. I just want to go over how this is unique. The way that it works is that you have your cDNA and you basically ligate adapters to it to circularize the transcript. That means that when the DNA polymerase binds and you do your sequencing, you can sequence the same circularized transcript a number of times to get several subreads or passes of the same transcript. And in the preliminary analysis, you basically collapse these into one circular consensus read. This allows you to parse out what might have been an individual subread error and instead identify single nucleotide variations with higher confidence. And then subsequently, the analysis that you're doing with this, or that we've been doing with the sequencing, is using the Scanty3 package. And I just want to highlight a couple of the features that are that we have been using when we've been doing this analysis. The first one is that we've been mapping to a reference genome to identify specifically which genome our transcript is coming from. As we are looking at human data, we can be able to use the publicly available human genome sequencing for this. Additionally, we are doing what we basically been calling an internal validation or in silico validation by including short read sequencing data, and I'll come back to this in a bit more detail. What I just want to mention is this is one version of extra data that you can include to feel more confidence in the isoforms that you're identifying. What can be recommended is adding matching short reads. We did something slightly different and added or basically mapped short read sequencing from different human organ donors to the transcriptome that we'd identified with long read sequencing to be more confident that the isoforms that we'd identified in this set of donors that we did long read sequencing on, we could also identify in different organ donors. I'm happy to take some questions at the end on more in-depth analysis, but that from now on, I would like to move into the specific data collection that we basically got from this analysis, from this data. So we did sequencing of 3 DRGs from 3 organ donors, 2 female donors and 1 male. We obtained a total of just over 3.1 million reads with an average length of 2,781 base pairs, which is close to what we were expecting for the average sequencing or average read length in the human transcriptome. We got really nice and high quality sequencing. This is a bit of a convoluted sentence. When we're talking about RNA sequencing, one of the quality estimates is basically the Phred score. The Phred score is a score of the estimate of accuracy of your sequencing for an individual base pair. So a Phred score of Q30, which is what we saw for 78% of our sequencing reads, indicates that we had a 99.9% estimated accuracy of the base pair call for those reads. So we were really pleased with that. And then we moved into our scan-T3 analysis, and the results that we are looking at are basically within this format. So the format is basically that you are comparing the transcripts that you identify, the isoforms that you identify, to a reference transcript indicated here in black. You can either have a full splice match where all of the splice junctions match perfectly. You have all of the exons that reference this specific reference transcript. You can have an incomplete splice match where you are matching the splice junctions of a reference transcript perfectly, but you might not have the full transcript recovered. And then we have these 2 novel categories. One is a novel isoform with a combination of known splice junctions. So the example that we give here is basically an isoform where the splice junctions have been identified with a specific pattern. In this case, the exclusion of this exon hasn't been annotated before. And then finally, this group, which was perhaps the one that we were the most interested in, was the novel isoforms with at least 1 new splice site. And in this case, it is the elongation of an exon, which may have implications for additional parts of the coding regions being added. So we were particularly interested in this group of novel isoforms. So to dive a little bit further into the data, we collected just over 900,000 reads per sample for a total of 152,264 isoforms. To look at that in a little bit more detail, we actually found that the largest proportion of genes had just 1 isoform, which was a bit surprising. That's not normally what we're expecting to see for the transcriptome diversity within a given sample. And we saw that this basically from the very high number of novel genes that we detected, which was perhaps a bit surprising. So the first clean of this data that we did was this inclusion of short read sequencing data, as I touched upon briefly. The way we did this was to basically add extra data to this data set, so basically map short read sequencing data from a different set of human DRGs to the transcriptome that we had generated with long read sequencing. The data set that we used for this in silico validation was a published data set from our group of really deeply sequenced DRGs from 3 different organ donors. Again, what we wanted to do in this case was basically feel confident that if we were calling something a novel isoform, it could be consistently characterized in DRGs from a number of organ donors. With this filtering, we basically narrowed it down to just under 94,000 isoforms identified. You can see that a lot of what we were filtering out was basically the novel genes, which is not surprising. And now we've got a graph that looks closer to this, where basically what we're seeing is for the majority of genes, we identify 6 or above isoforms for that gene, which is closer to what we were expecting, which is the story of incredibly high diversity of transcripts being generated through splicing. So, again, what we were incredibly interested in for this particular project was attempting to characterize the novel isoforms, so isoforms that hadn't been previously characterized, and then isoforms that were coding for proteins. So we did a filtering where we were basically just looking at these novel isoforms that coded for a protein or were predicted to code for a protein. And then we imposed some pretty stringent cutoffs. In this case, we imposed cutoffs of 2 transcripts per million, being necessary for us to call it an isoform for both the long read sequencing and the short read sequencing. Finally, once we narrowed it down to this group that had a novel splice site, so this were that last category that I was talking about, we were able to identify 763 entirely novel isoforms, so they haven't been previously annotated in our human dorsal ganglion tissue. These were a couple of additional questions that we had and were particularly interested in trying to address. The cell type expression of genes may be particularly relevant for a tissue like the dorsal ganglia, as it is a very heterogeneous tissue. So we have lots of different cell types being present within the human DRG and playing different roles. There's not just the neurons, which is these cells that extend along branches of nerves into the periphery. There's also different support cells. We were particularly interested in trying to figure out whether the isoforms that we were identifying, these novel isoforms, what cell type might they be coming from? The way we tried to address this was to characterize the expression pattern of those, the isoform that gene came from. We did this by comparing to this harmonized atlas paper, where they basically characterize the transcriptome, so the RNA expression within the cell types in the DRG. And then we looked at for a gene that we identified a novel isoform, what cell type were they most highly expressed in? And you'll see that for this bar graph, it really, really looks like that by far most of the genes that we identified that had a novel isoform were most highly expressed in neurons. So this doesn't necessarily mean that the isoform that we identified, the novel isoform, is expressed in neurons, but it points towards an incredibly high diversity of isoforms expressed within this specific cell type. An additional interest that we wanted to touch upon, of course, was this functional implication. The functional implication of a novel isoform, what role might they be playing? The way we initially looked at this was to basically look at whether we could assess just the isoforms that had a novel coding region. So may have a coding region which changes to what has been previously characterized. We had 514 of these isoforms. So I just want to touch a little bit upon or give a couple of examples of isoforms that we identified. Of those 514, there were multiple genes that were had previously been associated with pain. And once again, one of the ones that we found was WNK1. So this was this gene, WNK1, that I mentioned previously that we were interested in. WNK1 is particularly interesting because it has already had a isoform that's been characterized as uniquely expressed within the peripheral nervous system. Specifically, this isoform that expresses an exon titled HSN2. This isoform of the WNK1 gene has furthermore been associated with this hereditary type of neuropathy. So a gene that specifically has an isoform expressed in our tissue of interest, but also furthermore has been associated with abnormal pain sensation. In addition to this, quite close to the publication of our data, this really, really nice paper came out by PacBio, where they were characterizing the WNK1 isoform specifically expressed in the human dorsal root ganglion. And what they found, which was really interesting, was not just that they confirmed and really nicely characterized the expression of this peripheral nervous system specific isoform, they found that they could identify an additional novel exon within this gene that was unique to the peripheral nervous system. We thought this is something that we can basically validate with our data set. Really nicely, we found that we were able to do so. Now we'll go over this plot in a little bit more detail because I appreciate that there's quite a lot of information on it. Each of these lines basically represents an isoform that we identified in the human DRG. So each of these pp dot number is an isoform from our long-read sequencing transcriptome. And then each of these lines in blue is the Wnt1 or are the Wnt1 isoforms that have been previously characterized in the gen code V43 reference genome for humans. And each of these dark bars indicates an exon. So what you will hopefully be able to appreciate is that in quite a few of our isoforms, we see this exon, which is basically the exon that Matt Sapio characterized in his paper. It's this novel exon that was present in the peripheral nerve system in the Wnt1 gene. And this, again, it's not been characterized, so it's not present in the gen code genome, reference genome. Really nicely, we found that almost 20% of the expression of the Wnt1 gene expression came from one of the isoforms that express this novel exon. So to zoom in a little bit more, this novel exon is a 27 base pair insertion into the coding region of the isoform. So this is just a zoomed-in look at the specific sequence that is added. This is basically a screen grab from the UCSC database with our data populated in it. So you get access to all of the information that's previously been annotated about a given gene. And as you can see, our insertion exon is happening within the coding region, specifically what's encode the cytoplasmic domains. We furthermore predicted that this insertion may actually be inserting an additional phosphorylation site into this sequence, specifically a phosphorylation site that may be targeted by the GSK3 kinase. Now, it's actually been characterized that interactions between the GSK3 kinase and Wnt1 may play a role in neurite outgrowth. So it could be hypothesized that it's specifically the interactions between the GSK3 kinase and this exon in the Wnt1 isoform that's expressed in the human DRG that plays a role in outgrowth of neurites. One other example that I want to point out of a novel isoform that we identified in our data set was of the HNRNPK gene. So HNRNPK encodes an RNA binding protein. And I want to actually use, so hopefully this is a video that will play. This is going to show the interactive platform that we generated with all of our information in it. You can access it by using this QR code. But it's basically an interactive platform that allows you to search for your gene of interest and see which isoforms that we identified for that gene. So I'm going to be talking over and showing how we identified the HNRNPK novel isoform using this platform as an example. So this is basically the gene search feature of that interactive database. So it has this gene search side where you basically search for your gene of interest, and it will pull up this table of information that we generated for that gene. So you'll see that I'm searching for HNRNPK. And upon finding it, this whole table of information shows up. If you're fast, you'll see that's all of the isoforms we identified for this gene were novel. But you'll also see that we list all of the gene expressions of both for the long read and the short read sequencing in this table. If you want a visual look at the isoforms we identify, you can go to this Explore tab, which takes you to the UCSC genome browser database, but a version of it that we've populated with our data. So you can basically compare the reference genome, the gen code version, to each of the isoforms that we found. Once again, you can search for your gene of interest. In this case, I'm searching on HNRNPK. And you will see all of the isoforms as the black line are the ones that we identified in the human DRG. To be able to visualize it a bit better, what I'm doing here is basically zooming in to our region of interest in the specific gene. So you'll see that you'll be able to zoom right in, and then I will collapse the regions, so pack them all together, so you can see both the isoforms that we identified and the gen code versions in the same point of view. To zoom in a little bit further, we found a really nice variability between the isoforms that we'd identified and the previously annotated isoforms of this specific gene. So you'll see that not only do we have an elongation of a given exon in our isoforms, we also have an entire deletion of a different exon. In this case, it's the elongation of the shortening of elongation of exon 13, deletion of exon 14 in our isoforms. And this is consistent, as you'll see, for all of the isoforms that we identified for this given gene. So to briefly go over what that means, what we basically found was a consistent expression of a novel isoform of this gene, the HNRNPK, to be the only isoform that was expressed in the human DRG. And it contains an insertion as well as a deletion, which amounts to an additional 5 amino acids being added. We predicted this to not only exclude a specific site in an outlying loop structure, so in this specific structure, we predicted this to remove a phosphorylation site and potentially insert a different phosphorylation site within the sequence that could be targeted. Interestingly, we actually found this paper that associated HNRNPK with a Alpine syndrome. So mutations, loss of function mutations in the specific gene have been associated with this specific syndrome. And part of the experiences of these patients that suffer from this syndrome is abnormal pain sensation, indicating that this specific gene may actually play a role, especially with the location in the human DRG, in abnormal pain sensation. What I just want to highlight as well is that this entire data set is available on the Spark portal. This specific QR code will take you to that data set. What's really interesting about the data that's on the Spark portal as opposed to what's on the interactive platform is that I mentioned briefly earlier that we imposed some pretty stringent cutoffs. This was because we wanted to be confident that what we were calling a novel isoform was in fact consistently expressed in the human DRG. However, with these quite stringent cutoffs, we may have lost some isoforms that are simply more locally expressed in the human DRG. And if there is an isoform that your group is really interested in, has characterized in a different tissue, or for another reason feel confident that it might be true expression, you will find that the data set we've uploaded to Spark also contains a version that is not filtered with these very stringent cutoffs. So we have both the data set that is the filtered data set that contains this more stringent categorization of a novel isoform. And then we have the data set for each individual donor that we carried out long-read sequencing on, showing all of the isoforms that we identified in that donor. So I encourage you to check out both the interactive site, which will hopefully be a nice insight initially into the data that we curated. And then if you have more data that you were interested in exploring, or an isoform in particular that you really want to see if it may be present, the Spark data set is absolutely the way to go. So, and you can also always reach out to us, and we're more than happy to discuss the data that we collected. Which brings me to the summary. I hope that I've been able to convince you that long-read sequencing is a powerful tool to characterize isoform expression patterns. We at least have found it to be so. Some of the future things that we're going into is this idea of the isoform reference transcriptome that we've generated being a bioinformatic tool that could allow us to look at isoform expression patterns in other sequencing data sets. So the way that we validated the presence of a given isoform with short-read sequencing data, we essentially mapped a sequencing data to our long-read sequencing transcriptome to identify whether that isoform could be seen in that data set. And we think this is something that we could be doing for a multitude of other data sets, which could open the door to how we're identifying isoforms in a given data set. Additionally, something that we are incredibly interested in relates to just how heterogeneous of a tissue the human DRG is. So I touched upon the fact that it retains a multitude of different cells with different roles. The concept of the single cell transcriptional complexity is something that quite a few of us are working on in Ted's lab. We're using different techniques to address it. But I do strongly believe that the isoform complexity that we're going to be seeing will be on a single cell level as well. Finally, the functional implication of different isoforms and the novel expression of different isoforms in different tissues I'm sure is going to be opening a lot of doors for a lot of areas of interest of research. Which brings me to the end of specifically the long read sequencing portion of this talk. And for the final few minutes, I just want to give a little bit of a teaser for a different project that we're working on. So we are just putting the final touches of this on this project. In fact, the preprint for this paper just came out on bioRxiv this week. So this is a project where we were doing transcriptomic characterization of the cervical ganglia from patients, so not from human organ donors. We were able to recover this tissue from patients that were undergoing a specific surgery, the arthrodesis surgery. They were undergoing it for acute or chronic pain, and in that process, the cervical ganglion at the C2 level is removed. And we were basically able to then recover it and use it for experiments. What we did was use multiple different transcriptomic approaches to create a map of the transcriptome of the cervical ganglion from humans, which we believe has not been done before. And then we were additionally able to use these different transcriptomic approaches to characterize the differences to the transcriptome that is observed in patients with chronic pain. So as I mentioned, the preprint of this paper has just come out on bioRxiv. We're putting the final touches on it, but I encourage you to go give it a read. We are excited to receive some feedback on this. And the final touches on the data set are being finalized right now. We hope to have that data set online on Sparc within the next couple of weeks. So keep an eye out for that. I just want to say thank you to everyone who has worked on this and dedicated time and energy to this work. It has by no means been me alone, as well as to Ted Price, who's a fantastic mentor at UTD. To our collaborators at the Southwest Transplant Alliance. We couldn't do any of the work that we do without this collaboration. It's fantastically important to us. And then, of course, our funding bodies, as Sue mentioned in the beginning, we are funded by the NIH HEAL initiative, which is research towards finding novel targets for non-opioid treatments for pain. We are part of the HEAL Precision Network, so we're fantastically grateful for the funding that we received.